Hi, my name is Amos, and I'm the Chief Operating Officer of Cellect Biotherapeutics, a public traded biotech company that aims to enhance cell therapy. Cellect is developing products based on a unique and proprietary functional selection technology, which has the potential to deliver a broad range of next-generation cell and gene therapies that are reproducible, safer, and more efficacious. The opportunity here is in improved safety and efficacy of cell and gene therapies by improving reproducibility of cell products, improve bone marrow transplantation outcomes in allogeneic cell therapy transplantations, expand bone marrow transplantation markets by introduction of robust, reproducible, automated processes without need for a clean room, improve production efficiency and success for CAR-Ts and other similar processes like NK by making the processes more standardized. These CAR-T and NK products should have better efficacy and improved safety outcomes. There are also potential applications in other cell products, like in fast, high-quality expansion of mesenchymal stem cells. Cellect has an apoptosis-based functional selection technology. This technology is based on our findings that cells are differentially sensitive to CD95 receptor-mediated apoptotic signals. While most mature cells die following exposure to CD95 ligands, the same signal activates stem and progenitor cells. This technology is a simple solution for improved selection and expansion of cells. Cellect's technology is protected by a strong IP wholly owned by the company. This IP is validated by POC studies. Cellect's lead product is APOgraft. This is an improved bone marrow transplantation product. Bone marrow transplantation, or BMT, has been used for many years, and it is a very powerful treatment, but the main limitation with this treatment has been graft versus host disease, or GVHD. GVHD is associated with high morbidity and mortality, and this is why this powerful cell therapy treatment is currently limited to only few life-threatening indications. We have shown that APOgraft potentially increases bone marrow transplantation safety while maintaining its efficacy. And therefore, it can substantially widen bone marrow transplantation use to other indications, such as tolerance induction in solid organ transplantation and treatment of autoimmune diseases. APOgraft's method of action depletes mature and activated toxicity-causing cells, including T cells, B cells, antigen-presenting cells, and myeloid cells, reduces immune hyperactivation, meaning GVHD or cytokine storm, maintains stem and progenitor cells composition and function, maintains both short and long-term hematopoietic reconstitution, maintains graft's anti-inflammatory properties as well as graft's anti-tumor properties. APOgraft manufacturing is a robust and easy-to-perform process that can be easily integrated into current bone marrow transplantation procedures. APOgraft is based on very large and robust development data. Multiple batches have been successfully produced, with numerous in vivo studies and controlled tox studies showing preclinical safety. Clinical APOgraft batches were also successfully produced and administered as part of our first-in-man clinical study that I will refer to in the next few slides. Moreover, our apoptotic ligand residual APOgraft is about 250-fold less than the determined maximal tolerated dose found for that ligand in an IV dose finding and pharmacokinetic clinical study. Cellect's business has two pillars. One pillar is product development, where APOgraft is our lead product. And the other pillar is where we are committed to helping companies by enhancing their cell and gene therapies. In reviewing these two pillars, you can see that for APOgraft, we currently have 2 undergoing clinical trials on GVHD mitigation. These are conducted in Israel and in the US. One is in allogeneic PMT study, and the other is a haploidentical bone marrow transplantation study. Based on the safety profile from our clinical studies and very promising preclinical results, we are preparing a new indication clinical trial for induction of immune tolerance in solid organ transplantation, and future development could include for treatment of some autoimmune diseases. The initial APOgraft product was a longer manual process, but we have now developed an automated process that would reduce current manufacturing time and effort to about 4 hours using only 1 technician, and ultimately take APOgraft manufacturing out of the clean room. These improvements are planned to be introduced into future trials. We also believe these improvements make a paradigm shift in helping cell and gene therapy processes become more robust and reproducible. We continue to build data that supports our other business pillars of enhancing cell and gene therapies. We have conducted studies that show promising results in improving CAR T therapy and in fast, high quality and quantity expansion of mesenchymal stem cells. We have started conversations with potential partners across the spectrum where we hope to work with them to use our technology to help enhance their products and processes. Our preclinical studies of APOgraft in allogeneic bone marrow transplantation show improved safety of APOgraft while maintaining effectiveness. This is shown by improved survival compared to standard of care bone marrow transplantation, a significant reduction of graft versus host disease development and score, good engraftment, successful short and long-term immune reconstitution and stem cell functionality, and preserved graft versus tumor effect. Because of time constraints, I will not dive into the vast data that we have produced, and you are invited to read about our results in a recently published peer-reviewed paper on BMT's May 2020 journal, right in the link below. As mentioned, we have very promising preclinical results supporting the use of our APOgraft product in other indications, thus widening the use of bone marrow transplantation. These indications include induction of immune tolerance in solid organ transplantation and in treatment of autoimmune diseases. Some of the data was published in peer-reviewed papers mentioned in the bottom of the slide. For the sake of time, again, I will not elaborate on these results and will be happy to answer questions after this presentation. Cellect is in the process of finalizing its clinical study with a first-in-man dose escalation, 2-site study that is just about to finish. In this study, 11 patients were treated, and we have reached the treatment dose and safety profile that allows us to continue with phase 2 studies and with other indications. APOgraft has currently an orphan designation, and we are planning to submit an ARMA designation based on preliminary human data from our Israeli study. This designation will allow us to work more closely with the FDA and potentially accelerate APOgraft market approval. We are also about to start a US study in Washington University School of Medicine in St. Louis. This study of APOgraft is for the prevention of acute GVHD in haploidentical hematopoietic cell transplant recipients. The study's IND was already approved by the FDA and the WashU IRB, and tech transfer has been completed. Although COVID-19 restrictions have impacted the start, both WashU and Cellect teams are ready to start this study this year. In terms of differentiation, other cell therapy selection technologies rely on morpho-specific antibody-based selection resulting in non-specific selection of harmful and beneficial cells. In bone marrow transplantation specifically, these technologies cannot differentiate between functional subpopulations, for example, GVHD causing T cells and graft versus tumor causing T cells. In these technologies, it is difficult to maximize both safety and efficacy in the same product. They are also laborious and complicated processes, and lack of product consistency leads to unreproducible outcomes, a critical unmet need that leads to failure in clinical trials. Compared to these technologies, our APOgraft product selects unique cell subpopulation according to their functionality. Good cells are in, bad cells are out, forming product functional consistency. It negatively selects graft versus host disease causing T cells while maintaining stem and progenitor cells quantity and function. It improves bone marrow transplantation safety while not harming effectiveness. Graft versus host, graft versus tumor, and engraftment. And all of this in a significantly short, robust, and reproducible manufacturing time of about 4 hours. As previously mentioned, aside from developing our APOgraft products, we believe that our technology has broad application in enhancing most cell and gene therapies. Our apoptosis-based functional selection has shown promising results in preliminary studies in CAR-Ts, where we see potential for improved transduction efficiency by pre-selection of cells, and in reduced toxicity, meaning preventing GVHD or cytokine storm, by selecting cells after transduction. In mesenchymal stem cells, we see fast, high quality and high quantity cell expansion, X-fold expansion of stem cells throughout passages compared to standard expansion, better stemness, improved differentiation capabilities, enhanced immunomodulatory activity, and increased migration. There is a growing demand for high quality and quantity of mesenchymal stem cells that can be used in cell therapy treatments, as well as in cell-embedded scaffolds or polymer matrices. We have also started evaluating our technology's potential in NK treatment. As mentioned earlier, we are exploring discussions with a wide variety of partners, and we believe we could use our technology to help their product and process development activities. To conclude, Cellect continues to develop its APOgraft line of products while collaborating with current and potential partners that are interested in improving their cell and gene therapies products and processes. For more information, please contact us through the emails below. We'll be happy to answer further questions and provide more data. Thank you very much for your time.